For me, it's a pleasure to stay here with you, present my project aside on unraveling the regulatory role of elements in Arbuscular mycorrhizal symbiosis in tomato plants. For introduction, plant and fungi have evolved together from millions of years, and their interaction can be true, pathogenic, and beneficial interaction. In a few words, this interaction can be friends or enemies. Unfortunately, everything is not a beneficial interaction because we have fungi have pathogens, so cause a disease in the plants. An example of this is a late-life tomato caused by fluorescent. But in the positive side, we have beneficial interaction too. So it's an interaction that have a mutual benefit between plant and fungi. The example of this case is mycorrhizal. Continuing to the positive side. Thank you.
Just to pick up.
It's better now. Mycorrhizal symbiosis is a mutually beneficial association between plant roots and soil fungi belong in the Glomeromycotina. And in this interaction, plants got phosphorus and nitrogen from the fungi, and the fungi got carbon and fatty acids from the plant. We have several types of mycorrhiza, but I focus on two of them. The first one is the ectomycorrhiza. Is the type where for the mycorrhiza, 4 and 5 surround roots, but don't enter to the plant cells, to the root cells. An example of this is a woody tree, like oak or pine. And the other type is the endomycorrhiza, is the most common. We have 80% of species with this type of interaction. And in this type of interaction of mycorrhiza, the fungi form a structure called arbuscule. When they happen, they change nutrients. Yeah. But we have another type of mycorrhiza, for example, eukaryotes or orchidoids. And in this interaction, we have several key phases. The first is pre-colonization, is when the mycorrhiza interaction and between the plant and the fungi don't have a contact directly, but he sends signals each other to recognize and start the process. After that, we have the second phase. The root colonization is when the contact is a truth. And after that, we go on to the third phase. Is in arbuscular formation. Is when the fungi colonize and introduce in the root cell, and nutrient exchange happens. And in the final phase, extra-radical mycelium development is when the fungi go out again to the soil and explore new soil for transporting back to the plant new nutrients. Another important point to this process is the regulation of the mycorrhization. We have two important levels of here. The first one is the nutritional level, and a phosphorus is a limiting element, and we have two possibilities. The first possibility is high phosphate availability in the soil. Is when in this soil we have enough nutrient to know the plant, not necessarily the fungi for survive. Inhibit all root colonization from early stage. The second possibility is low phosphate availability. Is the opposite side. When in the soil we haven't got enough nutrients, and the plant activates all the response to recognize these fungi and or log signal to let the fungi penetrate. If you see the graph, we have two colors. The first is the white one. Is in the second possibility is the low phosphate availability, and in gray the high level. We see when no have phosphate in the medium, and if it's not see nothing, and it's more root colonization and more number of arbuscule in the root. The second point is a defensive response regulation, and one important element of defense response for us is MLO gene, because MLO is a protein with 7 transmembrane domain and associated calcium channel to modulate the signalase mechanism in the defense response. It's important for us because a loss of function of this gene confers broad spectrum resistance, effectively blocking fungal penetration and infection. If we see the photo, the wild type, we have a functional MLO gene, so the fungi colonize and penetrate in the plant and infect him. And in the opposite, we have a LO2, is a natural mutant variety that have a no functional MLO gene, so the plant don't let fungi penetrate and don't infect. But this is in general interaction of mycorrhiza, but it's the same in arbuscular mycorrhizal fungi interaction. Jacobs, Charpenter and his colleagues studied this process in barley, and you will see that in the first graph, where in plants colonized by arbuscular mycorrhizal fungi have an induction on MLO1 gene. So if we see here with an AM, arbuscular mycorrhizal fungi, who has a increase to the induction of the level of functional of the MLO. And on the other side, if we silencing MLO gene, affect to the colonization, and if we see it as an alpha or an arbuscule, we have a decrease of colonization and structure of colonization in the plants. This study is possible to have a study in wheat and Medicago truncantula. In the same paper, see that this collaborates, or this study, sorry, we have a specific clay to explain this type of interaction, as we say in this part. Medicago, wheat, and barley have a specific mycorrhizal host, but we question that it's possible in another species, and it's not in this clade. For example, tomato, Solanaceous rupesticum. We are studying this process in tomato, in moneymaker, is the variety moneymaker, and we do a colonization of arbuscular mycorrhizal fungi experiment with moneymaker, what I say, but and also the natural mutant. As I see, in moneymaker, during the days, we have an increase of the colonization percentage, but if we see OL2, the mutant, it's not colonize anything in all the experiment, but not is so rigorous because OL2 and moneymaker is not exactly the same variety. So it's possible that it's not a real control. So for this, we have a fusion line, and I back now. Fusion line is set the parents to possible the comparison the natural mutation OL2 and moneymaker each other. And we found LA1230 is a real control to OL2 is a parent, and we sent to sequencing and see is this variety not have the mutation, the mutant, and we see is another deletion. Sorry, OL2 is a deletion of 19 place of pair, and L1230 is not this deletion, this mutation. So considering this, our future research is with real control with the parent moneymaker and L2, play a bit with doing transformation for gene silencing and overexpression to the possibility to find a same phenotype silencing moneymaker and see the effect of the phenotype of the L2 or overexpress L2 to see the phenotype to the LA1230. It's only starting with this, but in a few months, I hope that it's more results to explain better this. Thank you for your attention. If you have any question. Thank you.
Can I ask you, the experiment with the mycorrhizal, did you use a commercial inoculum on one species?
Not, it's a commercial fungi. We have a, I don't know what I say, but we have a culture of fungi, Glomeromycotina, but and use this, but it's not a commercial. It's a one species. Yes, sorry. Coffee break. Thank you so much.